Hello, I'm Alexander Yarunin from the Global Oncology Diagnostics Scientific Affairs team in Astrazeneca, and it's my pleasure to welcome you to today's webinar. Today, we will talk about external quality assessment, why it is so important for precision medicine. And in the second part of the webinar, we will talk about increasing use of liquid biopsy for EGFR profiling. Wide access to targeted therapeutics for cancer treatment is one of the strategic imperatives for global oncology diagnostics at AstraZeneca. And one of the key aspects to achieve it is to ensure quality and accuracy of testing among all diagnostic laboratories. Participation in EQA schemes is essential for all laboratories to confirm the accuracy of their test results, to maintain quality standards, and often required for accreditation and strongly recommended in guidances, for example, ESMA. In addition, it gives extra confidence to healthcare providers and patients. Although tissue samples are generally used as a standard for molecular testing for cancer diagnostics, such as lung cancer, blood-based liquid biopsy provides a minimally invasive alternative and is coming now into wider clinical practice. And since the role of liquid biopsy is quickly evolving within the therapeutic decision-making, quality aspects of such tests are becoming increasingly important as well. Before we'll start, just a few housekeeping points. The next slide, please. Your microphone is automatically muted to avoid background noise. Please note that this webinar is being recorded. And please note that you can submit all of your questions using the Q&A window. Next slide, please. Your feedback is increasingly important for us, and it will help us to improve our future events. And if you would spend 2 minutes of your time to complete our survey after the webinar, it would be greatly appreciated. Thank you. Next slide, please. And it's my great pleasure to introduce you our today's speakers. It's Professor Sandi Deans, who is a consultant clinical scientist and the director of Genomics Quality Assessment, GenQA, which is a part of the UK National External Quality Assessment Service. Dr. Simon Patton, who is the managing director of the European Molecular Genetics Quality Network, and Professor Christian Rolfo, who is a professor of medicine and associate director for clinical research in the Center for Thoracic Oncology at the Tisch Cancer Institute. And in addition, he is the president of the International Society of Liquid Biopsy. Thank you for your time today, and I'm really looking forward to your presentations. Next slide. And next. To you, Simon and Sandi.
Thank you, Alexander, very much indeed. And welcome, everyone. Both myself and Simon are very pleased to have you join the webinar today, and we're delighted to have the opportunity to give you an overview of the results from the 2021 Liquid Biopsy External Quality Assessment, EQA, also known as Proficiency Testing Scheme for lung cancer. So next slide, please. So really we wanted to start with just to give you an outline of the objectives of the EQA scheme, this one in particular, which is slightly different from some of the other molecular pathology EQAs that we run, and also the format and how this is delivered. Then we're going to give you some results of the 3 cases that were provided, and then hopefully we can have a Q&A session at the end of the session after Dr. Rolfo's presentation. So the objectives of this EQA is to assess the testing accuracy, so the testing within the laboratories and the clinical reporting of EGFR variants in liquid biopsy samples. We started with EGFR in 2020 EQA, and also in response to participant demand, we have included the KRAS gene within the genotyping part of the EQA, which was optional because we're aware not all laboratories test for both genes. And really this was in the context of testing circulating free DNA in lung cancer samples. Now, the idea of providing this EQA is to help make improvements using a combination of assessment and feedback. And the feedback is given from an expert commentary. We have an expert panel with a lot of experience, a wealth of experience in the field of liquid biopsy testing, particularly in lung cancer. And we do this feedback both through individual laboratory reports, so that each laboratory receive a score determined by peer-reviewed marking criteria, and individual comments back from the assessors. And also we generate an EQA summary report, which again, it describes the format of the EQA, the expected results, and also a lot of the information that we gather from the EQA through the submitted reports. And we'll touch upon that, myself and Simon, later on in this presentation. So the format of the EQA is to assess the ability of the participating laboratories to, and there's 4 elements here. So correctly determine the genotype of the samples provided. So this really is the core of the EQA. Can the labs test the samples and receive the correct results for that sample? We also provide a clinical case scenario, because different interpretation of the results is dependent on what type of referral and the patient clinical management of that point of their treatment options or the stage of disease. So we interpret the results in response to the clinical referral, and we're looking for a clear, concise format that is not open to misinterpretation. We also want to see correct use of international accepted standard nomenclature, really predominantly HGVS nomenclature, and really the importance of this is to help use databases and other resources going forward, so the right result can be interpreted in the same context and we're all talking the same language. We also want to see appropriate and accurate patient and sample information and identifiers, and this is very important to ensure the accuracy that the result is going to the right patient, but also that we comply with some of the ISO standards, particularly 15189, whereby it's for medical laboratories testing clinical samples. And again, it's about the right result going to the right patient at the right time. So next slide, please. So to go back to the 2021 EQA, we had participation from 55 different countries, and here the graph shows the spread of the different countries, and I think you can say we have definitely got global outreach there. The predominant number of participants came from China, followed then by Italy and Spain, which is of interest. We had 352 laboratories register, 36 of them withdrew before the EQA opened, and we had a further 24 non-submitting laboratories for a range of reasons. So at the end of the EQA, we did have 298 laboratories testing the samples and submitting their results for assessment. So next slide, please. This is a schematic of the format of the EQA. Now, the EQA is provided by 2 of the EQA providers, EMQN and GenQA, but it's very important to highlight the fact that their EQA is run exactly the same way. It's a collaboration, and this schematic defines the different steps and how we do work together. So first of all, we open for registration. So registration is open to any interested laboratory. It doesn't matter where you're based or what type of testing you do, as long as you are providing a clinical service or setting up a clinical service for ctDNA testing in lung cancer. We source samples, and we'll talk slightly more in detail in a moment on the samples that we require for this type of EQA, but we source the samples according to our defined specification, and we're always looking to mimic exactly the type of samples and referrals that come into the laboratory. So we're not trying to trick anyone, we really do want to reflect your routine testing. And also to make sure that we know that the samples that we're distributing are exactly the genotype that we're expecting them to be, we perform validation on multiple methods in different laboratories. And really that's also to make sure that once we distribute the samples that we know that they can be used to obtain a reportable result. Then we have EQA distribution, and I think you can see here by the packages that with 292 laboratories, that can be quite a mammoth task, but they're distributed at the same time, and they obviously are delivered at room temperature, despite being liquid biopsy samples. They're artificial plasma material, and we've done a lot of stability studies to ensure that they are able to give that reportable result on receipt in the testing lab. We have online result submission through the two EQA provider websites according to a defined deadline, and then we also start generating the marking assessment. So this is according to peer-reviewed marking criteria, and this is in the context of the clinical case scenarios provided. So the genotyping is very clear what the results should be as expected, and we also seek clinical pathology and clinical scientist guidance on what the interpretation should be for those clinical cases. We then have the assessment period. Again, this is performed by an expert panel, and it is anonymized, so they do not know which laboratory have submitted which report, so thereby there's no skewed or biased assessment. There's a central collation of the results, and those are the data that we're going to show shortly. And then we, as mentioned, we provide an individual EQA scheme report to the laboratory and also an overarching EQA summary report. Now, because these EQAs have an interpretation element, interpretation can be open to subjective views. So we do have an appeals process. So if any laboratory feels that they do not agree with any deductions or any comments that have been applied through the EQA, then they have an ability to appeal. And again, these appeals are then reviewed by an expert panel anonymously, and a final outcome is returned back to the laboratory. The final reports are then issued, and we apply performance criteria just for the genotyping element of this EQA. Next slide, please. So in slightly more detail, EQA samples for the 2021 EQA were 3 custom manufactured artificial plasma samples that we were commissioned from SenseID. There was 80 nanograms per mil of cfDNA in 3 mil of artificial plasma. And they were validated by the manufacturer using ddPCR to ensure that they had the correct variant allelic frequency, and the samples were as expected before they were shipped to the EQA providers. So we, as I mentioned, we genotyped the EQA samples independently by 3 laboratories without their prior knowledge of the genotype before the EQA distribution. So here you can see here we chose Roche Cobas, ddPCR, and also a next generation sequencing, which is predominantly the most common testing methods that we know that laboratories use. And then the question was also asked of the laboratories that were looking for assessing the mock clinical referrals to create a scenario appropriate to interpret the genotyping results. So it's about adding that extra clinical interpretation onto your genotype report. So next slide, please. So snapshot of the testing methodologies, and I think this is the power of EQA. You can actually get an idea of what's happening on the ground in clinical service of all laboratories participating. And here you can see that there's 3 very strong methodologies that are in use, commonly in use. We have the real-time PCR, which Roche Cobas, no surprise there, I think is one of the most predominantly used methods, kits for real-time PCR. We also have NGS, and you can see the whole raft of different platforms and kits that are in use here, but you can see the peak right in the middle of the graph is all of the NGS participants linked together, so we have a large number there. And then from the middle of the graph there's a smattering of different other technologies there, with droplet digital PCR being the most commonly used. So next slide, please. So just a summary. I think one of the take-home messages from this EQA run that there's a huge number of variability of approaches and testing strategies. We had over 60 different methodologies being used, which is really quite astounding, and we don't quite see that as often in our other EQAs in molecular pathology. We had 173 laboratories using real-time PCR, and as I'd mentioned, Cobas is one of the most common, hence why we use that as part of our validation testing. We had 97 laboratories using next generation sequencing, and again, as I mentioned, using many different panels. Now, the most commonly used was the Oncomine Lung cfDNA assay, which was 14 labs, and then all others had less than 5 laboratories using them. So really there is a huge variability there in testing. And we had 44 laboratories using ddPCR. And I think one of the real take-home messages that we'd like to get across from this EQA run is that there are many laboratories there using kits that really are not suitable or on the market specifically for cfDNA testing. A lot of solid tumor panels are being used, or extraction methods, and we would really encourage you to review how you're performing your testing to make sure you get that optimal result for your patient. So next slide, please. I'm going to hand over to Simon, who's going to kick off the summary of the results from the EQA. I think you may be on mute, Simon.
I was on mute, apologies.
Brilliant.
Yes, thank you, Sandi, for your great introduction. I think what Sandy's already highlighted the methodology issue here, and I think you'll see when we go through these different cases that always some common themes around methodology and the use of different test technologies coming through in the actual results themselves. So I'm going to do case 1, and then I'm going to hand over to Sandi for case 2, and then come back to you for case 3. So looking at case 1, this was a male born in 1960 who had been diagnosed with metastatic lung cancer. And their original tissue sample had been tested and had failed on testing, and they'd been sent to the laboratory had been sent a plasma sample for testing for EGFR, which was the requirement, and KRAS was optional for those laboratories that undertook KRAS testing. And to give you a feel for the split between the number of laboratories, the percentage of laboratories doing KRAS versus EGFR, about 39% of laboratories on this sample tested KRAS. So clearly, not every laboratory yet is doing KRAS as a biomarker associated with lung cancer. Looking at the sample results, so we didn't expect laboratories to find any pathogenic variants in EGFR, but the sample did harbour a variant, a KRAS variant, a G12C variant at 6% variant allele frequency, and we were obviously expecting laboratories to pick up that variant. We were very careful, and Sandi again alluded to this in our material selection, to try and select variant allele frequencies that were detectable by most of the commonly used technologies. And certainly there's a very strong argument that any lab doing this type of testing should be able to detect down to way below 1%. But in real life, we see most laboratories drawing a cutoff at somewhere around 1 to 2%. For this case, for case 1, there were 8 genotyping errors. So that's 2.7% of all the samples tested by the laboratories. And there was 1 interpretive error, critical interpretation error. That's 0.3% of all laboratory results tested. So next slide, please. So looking a bit more in detail at the actual types of errors we found for this sample. The predominant number of errors, so 5 of those 8 errors were false positive results. So 1 laboratory incorrectly reported the presence of an EGFR exon 19 deletion, in addition to the KRAS variant they had actually detected. There were 2 laboratories that incorrectly reported the presence of another EGFR variant, so in this case L858R. 1 laboratory incorrectly reported the presence of an exon 19 deletion in EGFR, and also an L858R variant. And another laboratory incorrectly reported the presence of another variant in addition to the expected KRAS variant, so another EGFR variant. There was also 1 false negative result. So in this case, the laboratory failed to pick up the expected KRAS G12C variant. 1 laboratory incorrectly reported the KRAS variant. So they did, instead of detecting KRAS G12C, they detected KRAS G12R. And finally, 1 other laboratory did detect a variant in exon 2 of KRAS, but they did not provide any details on the test scope or the limitations of the test they employed. And again, this is a theme that we will see, we'll come back to. It's about how you report the sensitivity and limits of detection of the assays that you actually use. Next slide, please. So looking at in more detail at the interpretive error that was reported. So again, just to re-emphasize that these types of errors, the critical genotyping errors and critical interpretation errors, these errors are categorized as critical because they may have an impact on patient management. So in many cases, a patient wouldn't get treated with a drug, for example, if you've got the wrong variant. So we classify all of these critical errors as significant, and therefore they do, certainly in the context of the genotyping errors, lead to poor performance criteria later on in the EQA at the end of it. We don't provide poor performance at the moment on the critical interpretation errors. And in this one case, there was one error that we deemed to be critical interpretive error. So in this case, the laboratory incorrectly suggested that the result indicates that the patient would likely benefit from an EGFR tyrosine kinase inhibitor as a subsequent therapy. And of course, here in this case, the patient didn't have an EGFR variant, and they had a KRAS variant, the KRAS G12C variant. There is very good data from a phase 2 clinical trials that shows that a KRAS inhibitor targeting this particular variant, so G12C, has activity in non-small cell lung cancer. And on the strength of that data, the drug has been licensed by the FDA and the EMEA for approval of use in patients, and further phase 3 clinical trials are still ongoing. So we are completely acknowledge that the drug may not be readily available in all countries, but we do expect, and the assessors were very clear on this, that we expect laboratories performing KRAS testing to have an awareness of the trial data and to provide some sort of interpretation in respect to the use of these inhibitors. Clearly, these drugs are showing very strong promise, and it's very likely that they will be prescribed and readily available in many countries in the near future. So laboratories really should have an awareness of these types of drugs and the impact they may have on patient care. Okay, next slide, please. And just a couple of other common deductions that we saw from this particular case. So again, a common theme really across this case, but also the other 3 cases, overinterpretation of the result. So in this case, the overinterpretation of the absence of the EGFR variant. So as this sample didn't have the EGFR variant detected in the original sample, it's far too strong to suggest that the patient is unlikely to respond to or be a response to a second or third generation EGFR appropriate targeted therapy. In addition to that, there was no there's no recommendation for testing of a repeat sample or a tissue biopsy. So if you didn't detect it, you might want to check another sample or get a repeat sample to test. And again, quite a few laboratories were really very poor at providing sufficient information about the methodology used or the limitations of that methodology or assay that they actually perform. Again, a very common theme across this EQA, but also across quite a few of the other molecular pathology EQAs that we run together. Okay, I think that's end of case 1. I think we're going to move to case 2 with Sandi.
Thanks, Simon. So again, this was a referral for EGFR and essential testing and KRAS optional. So this male was presented with a persistent cough, shortness of breath, all the particular regular symptoms of testing required for lung cancer, and the testing had been requested to define management because there was not a possibility to have tissue testing available. So this sample was designed to have a very common EGFR mutation, the resistance mutation on L858R at 6% allelic frequency, and there were no KRAS pathogenic variants present in the sample. We saw a higher rate of errors compared to case 1. In this case here, we had 13 genotyping errors, so that's critical genotyping errors when a patient would have received the incorrect results, with 4.5% of participants. We also had some interpretation error, one error there, which again was 0.3%. So we'll go into these in a wee bit more detail now. So next slide, please. So we had, here are the false positive results that were reported. Again, we have 5, just like in case 1. So all variants, all sort of variable options under the sun here. We had a KRAS variant included, as well as the expected EGFR variant. We had a second KRAS variant detected by 2 labs, as well as the L858R variant in EGFR. We had a presence of a exon 19 deletion in EGFR and the T790M variant, as the actual expected variant was not detected. And then we had the incorrect reporting of the presence of the second EGFR mutation as well as the expected variant. So here you can see there's a huge range of different genotypes being reported. We saw predominantly false negative results being reported, and there was 10 laboratories who did this. And in response to really what Simon's point about the reporting the limitations and the actual scope of the test being performed, this really comes to in its own here. So these 10 laboratories failed to report the presence of the EGFR variant, and because we did not know the scope or the limits of detection of the assay that they were used, whether or not 6% would be expected to be detected, or they had no details whatsoever of the assay performed, then we were not able to actually mark these EQAs results, and we determined that they were false negative results, and therefore they were deemed as critical genotyping errors. Next slide, please. So moving on to the interpretation, I guess the good news is there was only one critical interpretation error, and as Simon stated, that we don't apply any poor performance on the interpretation at this point in time. This laboratory incorrectly stated that EGFR mutations are predicted a response to non-EGFR appropriate targeted therapy. So we, in the presence of EGFR variant associated with potential response to EGFR appropriate targeted therapy. And some labs we found that did not actually provide any specific interpretation of that result to the patient in that clinical case scenario provided. There were a lot of generic statements either in a footer or hidden somewhere in the report that were not tailored to that specific case, and really gave multiple different options of, if this result is reported, this is what the outcome is interpretive wise, and it was very unclear to find which interpretive comment was linked to actually that case. So again, another take-home message, please provide specific interpretation of the result for the case that you are answering, or the referral that you're reporting on. And we were very strict, and the assessors were keen to deduct marks just to make that emphasize that this is very, very important on a clinical report. So next slide, please. Simon, over to you for our final case, the EQA run.
Thank you, Sandy. I've got my microphone turned on this time, so we're sorted. So yes, the final case, case 3. Again, a male sample, so from a patient born in 1965. In this case, the patient, again, been diagnosed with metastatic lung cancer. But contrast to the other cases, they'd already had testing of their primary tumor, and they detected a exon 19 deletion in EGFR. The patient had already received first generation EGFR TKIs, and after a period of time the patient had relapsed and showed radiological progression of the primary tumor and further metastatic lesions detected. So in this case, the patient was being asked for testing of her plasma to see to identify any other further EGFR variants that might be showing in the ctDNA of the patient. So in this case, we weren't expecting to find any pathogenic variants in KRAS, but we had provided a sample this time with 2 variants in EGFR. So a classic sensitizing mutation in exon 19 EGFR, and a resistance mutation, a T790M. Now the selection of the variant allele frequencies again was carefully chosen here to reflect what we'd see in normal clinical practice. So it's very often the case that you see a much lower frequency, maybe 50% or lower of the resistance mutation. So the sensitizing mutation was at 4% and the resistance mutation was at 2%. But again, the selection of these variant allele frequencies was chosen to make sure that the majority of laboratories could actually detect the T790M variant at 2%. Like case 2, there were quite a high number of critical genotyping errors, and also a significantly high number of interpretive errors as well in this case. So we had 12 critical genotyping errors representing 4.1% of the participant laboratories, and 5 critical interpretation errors representing 1.7% of the participant laboratories doing this case. Okay, next slide, please. So looking at critical genotyping error, in this case, it was failure to report the variant within the limits of detection or scope of the assay, and or failure to report the variant but not stating what the limits of detection or scope of the assay was. And this was a very common theme, as we've already pointed out, for the other two cases, and particularly prevalent on this sample. Next slide, please. So looking at in detail again, now in this case there was quite a broad spread equally between false positive and false negative results. So there were 5 false positive results and 6 false negative results. For the false positive results, the majority of them were incorrectly reporting the presence of the L858 variant. Don't forget it wasn't supposed to be present in this sample, in addition to the expected exon 19 variant and the T790M resistance variant in EGFR. There was another laboratory that incorrectly reported the presence of 3 other EGFR variants, one of which is quite reasonably well known, so the CIS797R variant in EGFR. But of course, the validation data and the manufacturing of the sample, we obviously wouldn't expect to see any of these variants in that sample. Looking at the false negative samples, false negative results, the majority of those results were laboratories reporting, failing to report the deletion in exon 19, but reporting the deletion in exon 19, but failing to report the T790 resistance mutation. And predominantly the reason for categorizing these as critical genotyping errors is that they stated that the limits of their assay was less than 2%. So in theory, they should have been able to detect the variant and therefore reported it as well. There was one other laboratory that didn't report the presence of either of the 2 EGFR variants. And in this case, they stated the limits of detection for both the variants is less than the variant allele frequencies provided. So they were also categorized as a critical genotyping error. Finally, there was one more critical genotyping error relating to misleading information provided in the report related to the genotype. So in the results section, the laboratory had stated that there was no mutation present in either of exons 18, 19, 20, or 21 of the EGFR gene, but the interpretive proportion of the report actually stated that they detected the T790M variant. So there was a discontinuity between the reported result and the statement in the actual results itself, confirming they had found a variant in the sample. Okay, next slide, please. Looking at in detail at the interpretive errors we detected, again, a broad range. So there was one laboratory that stated that the T790M variant was related to a resistance in first and second generation anti-EGFR TKIs, with no mention of the third generation EGFR targeted therapies. Another laboratory, which obviously would have been relevant to T790M, another laboratory stated that the T790M variant is a frequent secondary somatic mutation that confers resistance, which is true. Furthermore, clinical studies have reported limited efficacy of particular therapy in patients with non-small lung cancer who actually harbored that resistance variant. A third laboratory stated that T790M decreases the efficacy of EGFR tyrosine kinase inhibitors. And again, we know that that's not strictly correct. And another laboratory stated that EGFR mutations are predictive of response to non-EGFR appropriate targeted therapies. Finally, the last critical interpretive error was that the laboratory stated that the plasma sample tested exhibited a sensitizing mutation and a resistance mutation, which increases or reduces, I think it should say, response to an EGFR tyrosine kinase inhibitor therapy. And again, that's just sitting on the fence. You're not providing any strong indication of whether there's going to be any likely benefit or not of that therapeutic response, of that therapy. So this was a challenging case, and certainly probably the most challenging case of the 3 cases. But overall, the number of errors is still relatively small compared to the number of participant laboratories. The take-home message, I think, across all the EQAs is actually that the number of errors were reasonably low, less than 5% across all the different categories. Next slide, please. So common deductions for this case. So a number of laboratories interpret the case wrongly in the context of the clinical context. So remember, Sandi was very clear about the real need to make sure that you read the referral. these cases are very carefully worded, and the genotypes very carefully chosen to try and reflect real, true clinical practice. And because the implementation of this type of testing is being used quite differently in many different contexts, it's really important to make sure you interpret the results in the context of the clinical case scenario provided. So that was a very common theme we saw across this and the other cases. And the other common deduction, or if you like, was the presence of the presentation of separate interpretations. So laboratories interpreting the results of the different variants they found separately and not putting them together in a cohesive way that really gave the overall result for that patient. So it means the reports are very difficult to read, and the clinician doesn't find it easy, or won't find it easy, to actually pull out the salient points from the report. Okay, next slide, please. So other interpretive issues, just to highlight a few, there's a few of them here. Again, common themes across this case, but also across some of the other. So common theme is not recommending further testing, because of course, clearly, if you don't find the variant, doesn't necessarily mean the variant's not present. It could do with the amount of circulating tumor DNA in the cfDNA sample provided, et cetera, et cetera. So it's really important to make sure there's some recommendation for further testing. No variant results, but no variants detected or results, but presence of the original variant. No variant results and no original variant detected. Over-interpretation of what happens when you don't find a variant. The use, and this is a common theme that we see in many of our EQAs, the use of the terms positive and negative, which can be very easily misinterpreted by a clinician as to meaning something completely different to what you're trying to say. Again, limited scope of the test applied. So we've already seen plenty of evidence in all those three cases about laboratories that aren't stating what they've really tested or how they've tested it. And no interpretation of the result with respect to EGFR appropriate targeted therapy. So again, certainly in the context of resistance mutations, laboratories should really be thinking about the third generation TKIs. So if you're interested in reading more about the details, then please do obviously make sure you read the summary scheme report, which has a really thorough and completely comprehensive report on the outcome of the EQA and the way the laboratories performed, and goes into all these themes in much more detail. Okay, next slide, please. So finally, some acknowledgements, and it would be remiss of Sandi and myself not to mention, firstly, the two people from our organizations that have done all the work. Sandi and I have done none of the work here. These are the people who've really done all the hard work. So Jenny and Mel from a GenQA and EMQN, and also to all the assessors who helped us do the assessment, as Sandi alluded to earlier, the experts. These are the people who really know the stuff. And without them, we couldn't provide these EQAs to the same level, high level and quality that we expect of ourselves, as well as the laboratories. And finally, obviously, to you, the laboratories that participated and took part in the EQA. So I think that's it from myself and Sandi, and we're going to move on to Dr. Rolfo, I think, Alex.
Yes, thank you, Sandi and Simon, for the presentation. And just to remind everybody that we will have enough time at the end of the webinar, so put your questions into the Q&A window. We will address all of them after the webinar. And now to you, Professor Rolfo. Thank you.
Thank you. Thank you very much. It's a pleasure to be here. And actually, for an oncologist, speaking with pathologists is really a dream. So I thanks to Professor Deans and Dr. Patton for the great insight of this. I think it's very interesting to have this data of this survey, because it's giving the reality what is happening around the world in the testing. So that's are my disclosures. And I will try to summarize a little bit some concepts in liquid biopsy as an oncologist that I think are very important for pathologists to take in consideration. We know that in the last years, lung cancer was changing completely in the landscape of therapeutics, because we was passing from analyzing drugs or putting drugs in patients to see which one of them to respond, to determine molecular profiling on patients and get another biomarkers or predictive biomarkers, and even changing our vision of how we are not only treated, but also how we are designing clinical trials for these patients in the future. Obviously, we are talking today about liquid biopsy, but we don't need to forget that it was departing. And actually, as a pathologist, you are departing from the tissue. And we know that there are advantage and disadvantage. One of the disadvantages that the tissue have is the lack of capture of the tumor heterogeneity. And I think this is important right now, because if we are talking about driver alterations, we need to know what happened also at level of the clonal evolution of the disease. Obviously, it's really important to have the advantage of pathology as information assessment, also of some other biomarkers that we are using now, for example, of immunotherapy. But one of the advantage of liquid biopsy when we are using platforms, so when we are trying to get a big information, is that we are capturing this heterogeneity. There are also the possibility to be repeat over the time, and this is fantastic for our patients in terms of monitoring. And some of the disadvantage, obviously, and that could be part of the false positive or false negative that could come from, for example, from clonal hematopoiesis. That is a big still problems in some of the platforms and that we have, because we don't have actually 100% filter that is avoiding completely this problem, with exception if we are going to other methodologies that we will see later on. So I was talking about heterogeneity, and I think it's really important that we internalize the concept that there is not only the heterogeneity in the sample that we are analyzing, but also in the intra-patient heterogeneity. And this is a very important concept when we are treating patients with TKI, because we are not only looking for one driver mutation, we are checking for the co-mutations that can impact in the outcome of these patients. And also in case, for example, nowadays, we are going a little bit further and analyzing the liquid biopsy of the CSF, for example, in patients who are progressing in the brain exclusively. The liquid biopsy could be or the ctDNA analysis could be applicable in all the journey of the patient. We started with a lot of data and robust data in metastatic disease. And there, unfortunately, even if we have a plethora of drugs that were approved recently by FDA, the testing rate, and this is an analysis that was presented in ASCO last year from my lung consortium, that is a consortium of practice in United States, and you see that 50% of the patients are tested. And we are talking here about the most common targets. So we are not talking even about a TRAC or red fusions. And we have also the data coming from disparities. If we are applying that to, for example, African-American population, we are going to 39%. So we are doing as an oncologist very badly in asking that, but also in the creation of flowcharts or the flows in the institutions to get obviously reflect testing in some of the patients that will be obviously helping for the detection. This is the rate of the increase of the detection over the years, and is not comparable at all with the approval that we had for the new compounds driver for driver alterations. One of the studies that I would like to refer is the Nile study that was like this one, was also real world data. And in this analysis, they include patients who have tissue and liquid biopsy to see what happened in this core concordance, but also how many patients are able to really have a complete analysis of the 8 guideline recommended biomarker at the time of by NCCN guidelines. And you see here that several of the patients have the black spot is the quantity non-sufficient. And if you see the concordance was really high, 98.2%, 100% predictive positive value with the ctDNA, and high sensitivity for the 8 guidance recommended biomarkers, that was 80%. One of the things that is really important is the reduction of the ctDNA results. So, and that is what we are doing actually is a blood first, because we receive the results several times before the tissue biopsy. And when we are using this complementary approach, the increase of the detection is around 48%. So the blood first approach, that when we receive the results, is obviously something that we are applying. And actually, there are other trials like this. This is the BFAST trial, that is a prospective analysis checking for the blood as a only source of testing for these patients. There are several cohorts. Actually, it's very dynamic, this design. Some of the cohort, like the red, was closed because the drug was not the correct one to have for this driver. But in the case of ALK, for example, the data that we have are very similar with the pivotal trial that was using the same drug for tissue analysis. So it's very important that we can test our patients and have this opportunity to treat them. In 2018, we published by ILCLC the first statement paper about liquid biopsy, and we gave to liquid biopsy a secondary role after the tissue biopsy was insufficient. And then when we are talking about patients with advanced disease progressing to TKI, we gave to liquid biopsy the first opportunity for detect the mechanism of resistance that was involved in this progression. Nowadays, we have a new statement paper that was published in October last year. And beside the sequential approach that we gave in 2018, we are talking now about complementary approach, similar to what I was talking in the Nile study, but also the blood or the plasma first approach that is really what I say is happening when we are using the complementary approach. But also we include the monitoring, that here is very important. Several of the vendors in the United States are including right now on a kind of combo that allow to the doctors to do for the same approval, one test in the beginning and 6 to 10 weeks after the treatment with TKI is starting, to see the dynamics of the DNA as a predictive biomarker for outcomes. We was talking, and I saw one of the questions here in the chat, that how we get the allelic fraction. Obviously, that I will let Dr. Deans and Patton to answer that. But what is important that we are using this quantity of DNA present in the sample that is obviously related to the burden of the disease as a predicted biomarker. And this is, for example, an iconographic representation of the BAF. You see here in the top of the list. This is the BAF from the very beginning. In a journey of a patient, that was a patient that was treated. This patient having an EGFR mutation, common mutation, with some companion mutations like TP53 and EGFR amplification that are obviously not a good companions, that I call the troublemakers. This patient was responding very well to the first treatment that was a third generation TKI, and you see the reduction of the BAF, really a very nice reduction, complete response in the almost complete response in the lung, complete response in the brain, but after a very short time of 2 years, due to this companion, and this is important why we are looking next generation sequencing panels. Because we have this information. This patient was having a progression with a small lesion that was a 797S EGFR mutation that is a common mechanism of resistance for third generation TKI. This patient received radiation, but in a very short time, you see here from July to August, you have a big progression. And we did at that time, due to the position of the mutation and a TKI of first generation with a very good response. These areas that you see here are related to radiation and pneumonitis. But obviously, after 5 months, we was pushing other mechanisms of resistance typical for first generation TKI, and that was the T790M that arise. This patient have multiple nodules. We gave chemotherapy, as you see here, and reduction again of the allelic fraction variation. So this is a very nice example how we are customizing the treatment patients according to the monitoring of these mutations and the clonal evolution. We was talking about BAF, and this is a waterfall plot showing a very nice response in almost the majority of the patients, including complete response. And what is important from this study, that the majority of the patients have an BAF that was very, very low, because this is a common confusion that or mistake that the oncologists are doing, thinking that they need to have a percentage of BAF that is high for response to the patients to the TKI. And that is not really true. As far the patient have a BAF that is presenting the mutation or confirming the mutation, they can respond very nicely. These dynamics of the BAF we are using not only in the TKI, as I said before, but also we are now moving to the space of immunotherapy, and patients who have ctDNA positive or increase are going worse than the patients who have a decrease, and in both scenarios for overall survival and progression-free survival. And that is used also for other mutations or aberrations like you see here in ALK translocation. Obviously, all this tsunami of genomic information is really to decipherate and have a good interpretation. So we are working now with tumor boards, and obviously in the tumor boards we need to have some rules, and we are applying some level of evidence like these ones that is OncoKB, and I invite you to go for the website. This is a level of evidence created by Memorial Sloan Kettering, and you have a level of evidence one, for example, is the typical case of EGFR with the FDA recognized biomarker and drug. But we are going to other biomarkers on other levels, for example, level 3A, and this is the case that there are compelling clinical evidence, but neither the biomarker or the drugs are approved for that indication. And that is the case, for example, of ATM mutations and PARP inhibitors, that, for example. Another levels of evidence that we are using in Europe, there is the SCAT. This is the European Society of Medical Oncology, ESMO, create this it's a little bit more dynamics, I need to say, but it's a little bit more complicated for a clinical oncologist to be applied. Other level of evidence that are really simple is a simplification of the level. This is the MD Anderson, that is only 3 levels, but it's also very easy to apply in the clinical practice. We did an analysis using the OncoKB in patients who go for phase 1 trials, actually, and we saw when we are applying the tissue and the liquid biopsy, even if we have levels 3A, patients are able to enter easily in the clinical trials and have at least a rationale for these phase 1 trials for patients when we have these discussions. Another scenario that we are now talking and is really interesting, and we will see a lot of things going on in the near future, is the minimal residual disease. And here approach we will see, and I will not enter here in the details, but we will have several approaches that are tumor-informed and tumor-uninformed approach. So be prepared for this near future. We have several clinical trials going on. And the idea is to obviously to monitoring these patients, and here with EGFR we will have a very important activity, because with the 3rd generation TKI, actually in new indication for patients who have a resection and even an adjuvant opportunity, this will be important to tailor and monitoring these patients over the time to see when they are relapsing. The same concept in adjuvant is arriving to immunotherapy, and we have here, for example, this is one of the trials that we are using an immunocheckpoint inhibitor in adjuvant setting, and we see that patients who have ctDNA positive are going worse than patients with ctDNA negative after the adjuvant treatment in terms of disease-free survival, and so we will have a lot of things going on in the future. And early detection is the new holy grail in liquid biopsy, and there are obviously the idea to increase the detection of the screening programs with low-dose CT scan, and we are also using tumor-informed and tumor-uninformed. I was talking in the very beginning about the clonal hematopoiesis is a problem here, but when we are using, for example, methylation of the DNA or leukocyte analysis as well, we are avoiding one of these problems, and we saw that there are some data very interesting in terms of specificity, very weak still in terms of sensitivity, but very promising, I will say, for the future in detection of drugs, of lung cancer in the very beginning. So this is a slide that shows that I'm a dated oncologist, but in the future, if we have the opportunity to ask to somebody who is coming back from there, the future of liquid biopsy seems to be very cool with all these other analytes in place, and we can be able to use them, and we cannot wait for be there. But I hope that the person who is coming from the future is not telling us that there's still people not testing, and that will be very frustrating. So with that, I close, and I invite you for the International Society of Liquid Biopsy annual meeting that will be in Florida this year in Miami in 21 and 22 of October. And I'm happy to answer the question with Dr. Deans and Patton. Thank you.
Thank you, Dr. Rolfo. So we have some questions in the chat. So I welcome Professor Deans and Dr. Patton to answer the ones which are related to your topic.
Thank you, Alex.
Okay, Alex. Simon, do you want to kick us off then?
Yep. So we've got a question about, we see there are different false negative and false positive results, some of which could directly affect clinical management. Could we know what methods that generate the most errors? And I think we've also got another question, which is very similar to this. Are there discrepant results, wrong results, interpretations related to a single gene test or to next generation tests? I've just checked the EQA scheme report, and this year we haven't provided a breakdown of the methodologies that cause and how the errors are distributed across the different methodologies. It's something we quite often and frequently do if we find it, particularly if we find any clear trends, and EQA is very good at detecting these types of errors. We haven't done that, and I haven't got the data in front of me, but it's something we could probably break out if we need to, and we'll certainly be looking at doing that in the future. But yes, we can do that. We just haven't done it in this year's EQA.
Simon, I think the reason for that is there was no actual trends. We often, through the EQAs, as you say, we can detect maybe some nuances or issues with particular kits, and we normally feed that back to the commercial companies themselves and the participants using them. This year, I think, as we see, basically when clinical testing's getting embedded, just like liquid biopsy testing is now, there's no real themes and issues. It's really just business as usual errors. I think you can get different, particularly for interpretation, that tends to be something completely different and tends to be more educational, regardless of what type of methodologies that is employed within the laboratory. But also we see different types of errors for single gene tests and for NGS panels. So they all have errors, but they maybe are generated from different issues within the laboratory. We also see quite a lot of sample swaps happening within the laboratory as well, from any point within sample reception, DNA extraction, testing, and even just submitting results or putting the results from the platforms into the reports. So I think we don't want to get sued, obviously, by naming and shaming any particular kits or methodologies, but I think the take-home message there is just know your limitations of the methods that you're employing very, very well, so you can report them accurately.
Yeah, I would agree entirely with all of that, Sandi. And the other thing I would add, obviously, is that from the slides, you can see that there were 60 different types of tests used.
Absolutely.
And so any error we would see would be diluted down by the fact there's a sheer huge number of different tests there. So it would be very hard, as you say, to establish some really clear trends. But you're quite right. In any circumstance where we see a really clear trend, we will nearly always go back to the manufacturer, for example, and to have a discussion with them about methodologies, etc.
And actually, if I can say something, that was a problem for us when we was doing the guidelines, because trying to be international and give an international view of all, obviously we prefer next generation sequencing, and we say that, but we need to be aware also that the realities in different countries is completely different. But if we can give some recommendations also for example, the T17IM, that is still a problem in several countries, that we don't have third generation TKI approved. Some of the methods that they are using, like the COVAS version 2, is really weak in the detection of this mutation. We are talking about 59%. So it's having a plethora, as Simon say, of different testing is giving a variability in the results. But obviously, if we are focused or we are doing a sub-analysis of your results for the really high sensitive platform like next generation sequencing or digital Doppler PCR, the result will be obviously more higher, no?
Absolutely.
Absolutely, yeah.
So Simon, we've got another couple that are sort of linked as well, and that's about allelic frequency. So how do we get the variant allelic frequency? Which variant allelic frequency is most commonly used for estimation of the variant content in a large background of non-tumor DNA in cell-free DNA analysis? Why is this module requesting the variant copies per mil, assuming per mil of plasma? And also, what is the limit of detection suggested for real-life liquid biopsy testing? So all sort of themes there. Variant allelic frequency can be picked up through your bioinformatics, if you're particularly using NGS. So I would suggest you speak to your bioinformatics team to find out how you get that information. It's also very important to interpret the result, whether or not, particularly if you've not picked up a variant there, about what your limit of detection is, and that should be determined before you start any clinical testing. I think for what happens in real life, you have to go back to what the clinical question is that's being asked. Dr. Rolfo mentioned about when you're doing monitoring residual disease. Obviously, that is a slightly different question if you're doing upfront diagnosis, because you can't get a tissue sample, for example. For monitoring, you want to go down to, I would say, about the 0.1% mark. But I think obviously if you're doing a diagnosis, you don't necessarily need to go down as low as that. It depends on what your clinical team will act upon. We know that some ctDNA testing is happening, but clinicians won't act upon it until there's actually some sort of radiological evidence there, or there's a relapse sort of starting to show clinically. So again, I think it's about that, speaking to your clinical team, understanding why you're doing the testing and how it should be reported, and your assay should reflect that. And then finally, Simon, I'll come to you to add on to that about we're expressing variant copies per mil of plasma. And this, we know that many ways of determining the amount of mutant or pathogenic variant within samples is available, and labs use different ways to determine that. We have used copies per mil because we are using artificial, commercially made plasma specifically for us, and that is the way that the manufacturers are determining that variant level. So really it's a nuance of the EQA scheme that is the mechanism that we have used to determine what we want from the commercial company, and then we portray that across to the labs and also supply how much DNA is actually in that plasma sample. Simon, do you want to add any of that? I know it's all sort of packed together around variant allele frequencies.
Yeah, I would add to, particularly to the last point, that I think this is an interesting question, and it's actually very assay specific as well. And the way you establish the number of copies of a variant or the variant allele frequency itself is very dependent on the test technology you use, but also the validation of your test, and should be, in many cases, it's probably appropriate to actually try to establish both as part of your limitations of your test and assay specificity. So it's not easy to do. We accept that, and as Sandi said, the reason we report it in the EQA in this particular context is primarily because that's how our specification and manufacture from our manufacturer came. So we're using the validated assay that they use, that's done to an ISO specification as well, as if you like, the starting point, the truth of the number of copies present in that sample. And as it's artificially manufactured, it's reasonably easy for them to determine what that is. The other thing I'd like to add is to what is the limited detection suggested for real life tissue biopsy test, liquid biopsy testing. And again, I think this is a very interesting question, and I think hotly debated perspective as well. And Dr. Rolfo, I'm sure, will have much more to add to it than I will. But really, I don't think at the moment, the test, this approach to this type of testing using liquid biopsy is still, I would argue, in its infancy, and how it's implemented in clinical practice is still not very cohesively done. And there is I think, in my opinion, more guidance that could be given to laboratories and the clinicians as well about when it's appropriate to use it and what levels of sensitivity, etc., that you should be using. So I think as the technology evolves and the pathways, the clinical pathways evolve, we'll have much more feel for what this limit of detection really should be. But as Sandi said, if you were doing minimal residual disease, it should be pretty low, but clinical diagnostics maybe doesn't need to be quite as low as that. Okay.
Maybe I can say something about that. So obviously for the metastatic disease, it's really clear with we are talking about vendors because there is here a big difference. What happened with vendors with next generation sequencing and what we are doing in-house. And here in-house, we have several other problems that we need to face that are really standardized in vendors that are, for example, the pre-analytical conditions. So that could be given an important variability of the result that we have. So in terms of the vendors, what we have approved by FDA here, that is really known. We have a limit of detection that is above that we can calculate. And it's really clear that we can take even alterations that are 0.01 or 0.1% of the BAF. This is a really nice sensitivity. If we are moving to, like you say, Simon, in minimal residual disease, there we have still application of other kind of platform. So we need to be very careful when we are discussing about in-house and vendors because sensitivity and conditions and everything are completely different. So it needs to be, and actually that was one of the problems of in the survey of the American College of Pathology in general in test for tissue and liquid, how we are reporting, how we are doing the pre-analytical condition, quality of controls for the in-house technology. So this is a really interesting point of consideration.
We have two EQA specific questions remaining at the moment, and then I think Dr. Rolfo, there's quite a few for you. So if we just do those two first and then we hand over. So we have why, as a deduction, evaluated a recommendation for new testing of a new sample. It is known that the level of cfDNA changes. I think that's a misinterpretation of the presentation. So we said the deduction was applied when there was no recommendation for new testing or a new sample taken. So again, it comes down to over-interpretation when no variant has been detected. You don't have that reassurance there that you've actually tested any tumor DNA. So really the idea would be you would ask for a repeat blood sample to repeat that testing, or look to see whether you can get a biopsy or if there's another tissue sample available. Simon, do you think that's the misinterpretation there of that question?
Absolutely, yep.
Yeah, and then Simon, one final one to come to you that was about the EQA. It says, didn't hear at the beginning of the meeting, but we mentioned that there was an unsuitable kit used by a lab in Egypt. I don't think that's the case. We've not touched upon anything linked to any countries.
No, nor unsuitable kits, as we discussed just not very long ago.
Yeah, thank you. Don't want to misinterpret anything. Alex, will we come back to you if you want to look at some of the other questions?
I have one question to you, Dr. Deans and Dr. Patton. From your experience, have you noticed any trend in failure rates when you're comparing large CGP panels versus more targeted panels with a smaller number of targets in it?
Not particularly, no, Simon, go on.
I was just going to say the same thing. No, I mean, I think we kind of touched upon it earlier on, is that there's such a large number of different tests, Alex, and that we didn't detect any common themes.
Okay.
But I think maybe it's because we're not asking for multiple targets to be reported. So we wouldn't know if you couldn't generate data for other targets as well as EGFR and KRAS. So that leads on quite nicely to some of the other questions that have been submitted. And that's about fusions and detecting other clinically relevant variants, particularly in lung cancer. And both EMQN and GenQA are currently designing, in collaboration with IQN Path, a multiple biomarker EQA in liquid biopsies. So that's looking at your panels with larger and including the fusions in there. We also know that the NTRK fusions are something that's becoming very more pertinent for testing across the different cancer types and other markers, for example, in breast cancer setting. So that's sort of the next generation of our EQAs that will be coming out. So we will be putting a call out to our laboratories to see if you want to participate in those EQAs coming forward. So again, it's if we don't ask for multiple targets to be reported, we don't know if maybe they're failing as well, in particular the fusions. So we've always got a challenge ahead of us, I think.
Although
Definitely.
undoubtedly, I also expect that it'll be much more challenging. I mean, it's a much more difficult ask, isn't it, as well? So it's going to be very, very challenging as a field of testing, I think.
Yeah, in some of the fusions, like you mentioned, NTRK, that is a really important challenge for liquid biopsy. We published the first data of only one gene, that was the NTRK1, because put in a platform, actually for a vendor, but we are not talking in-house, with all the possibilities of fusion partner, because NTRK is very promiscuous in terms of partners for fusion. So it's really difficult. You need to have a platform with a very high sensitivity, and now we are arriving there. But for that will be a challenge for in-house technology, I think, because you need to have quality controls for every fusion.
Yeah.
So this is a big point. I see here that there's a question: can we determine TMB by ctDNA? And yes, we can, but be careful because obviously it depends on the platform that you have. It's not the same to calculate with 76 genes or with a whole genome sequencing. So that's the tricky part, and I'm not really like it to see at the moment the only one number in TMB. I think we need to be a little bit more smart to find which kind of mutations are really involved in the presentation of neoantigens, for example. But it seems that the BAF or the MAF are much more sensitive than TMB in the last results that we saw in clinical trials. Alex. Oh, sorry, Sandi.
I was going to say there's one, and I'm quite intrigued to hear your answer to this. This is more pathology based. So somebody has asked, can we predict with any confidence the histology of a cancer like non-small cell lung cancer using liquid biopsy?
I was trying to avoid that in a
I thought you might be.
No, I can say obviously pathology is in that we cannot really replace. But what we have, for example, in EGFR, and we saw in some patients with EGFR, as a mechanism of resistance could be the transformation to a small cell lung cancer. And that obviously with the liquid biopsy cannot be predicted, but can be, I would say, seen as a tendency. For example, if we have some companion mutations, and that is the case of retinoblastoma 1, RB1, and TP53. If we have a patient that from the very beginning have these mutations as a companion of EGFR, this patient have a possibility to develop that. And if we are losing these mutations over the time, that could be an indication. The contrary way, gannancy of this mutation was also related with the histology of a small cell lung cancer. But this is just one interpretation. We need to have the pathologies. What happened is that in some patients, we don't have histology. And that is happening also in other scenarios, that is in early stage, when we have patients that we don't have histology and we need to treat these patients with SRS, for example, with radiotherapy, and we don't have histology. And that is the common question that we have in the tumor boards. Can we have an idea what is happening with this patient? Is it a cancer or not? We don't have this data, unfortunately.
I ask you, just we're seeing a lot of referrals for liquid biopsy coming in, particularly following COVID, and with our huge, particularly in the UK, a huge backlog of cancer referrals. Are you saying that, you know, are you getting a lot of referrals coming in without that histology because you haven't got a tissue to assess, first of all? Do you have that triage process in place? Yeah,
so we see, unfortunately, you know, all patients that have a very big EPOC or COPD, or they have a very small lung nodule that are really difficult to arrive with, even with a bronchoscopy or in a transthoracic puncture. So it's really difficult to get there. And sometimes we say, okay, we will treat this patient because have all this nodule was following and is growing and have the radiological characteristics. We are using now radiomics as well to identify some of features. But even though we don't have the histology, so I think this will be a really nice scenario where liquid biopsy can give an important contribution.
Yeah, and we've just started a trial in the UK in NHS England about looking at use of liquid biopsies where there's not a confirmed diagnosis yet. So that will be very interesting to see the results from that.
Yeah, and actually the methylation, for example, there is a company now that was approved by the Medicare, that is the company for reimbursement, not still approved by FDA, but is using methylation. And we saw in lung cancer, the sensitivity is for stage 1 is 21% with a high specificity, but the sensitivity is low. So still we are missing several patients. That will be a problem. So I think in the future, we will see a lot of multi-omics approach to identify these situations. So I hope so.
We have one last question, I think, by the looks of it. So this question is, I think Sandi and I should be able to take it, but Dr. Rolfo, if we can't, we're coming to you. What is the recommended method for the pre-analytical QC before DNA analysis? And I think my response to this would be that our response from as an EQA provider is that we can't obviously make a recommendations of the particular method you use. And we certainly know from this EQA that there are many different methods being used in the pre-analytical process of the extraction of the DNA, if you like. But one thing is for sure is that you need to be using an assay that has been validated for cfDNA. And there is good evidence from several other EQAs we provide of labs where they're not using an assay, a pre-analytical step that's been validated for cfDNA, where there is a higher chance of getting errors because you're not extracting the right amount of DNA, or it's the wrong fragment size, etc., etc. So I think it's true that you need to be making sure you use the right type of assay for the pre-analytical step. Would you not agree, Sandi?
Absolutely. I think that's the take-home message, isn't it? That you tailor what your assay is for what the end answer that you're trying to obtain. As you say, we perform an EQA for DNA extraction from blood and tissue types, not for liquid biopsy yet. And we see huge variability between labs trying to determine concentration and quality of the DNA, even using the same methodologies. So there's huge variability out there. And I think it's about making sure that you validate what you do in your lab gives you the right result. It's, it's arbitrary in a way between labs. It's as long as your processes are fit for purpose.
Yeah, there are also some, in this regard, there are some societies or groups that are working in preclinical analytical condition. And this is the case of BloodPack, that is was coming like a moonshot initiative for the government in the period of the government of Biden, and then became a group, Barack Obama, and became a group of stakeholders putting together some guidelines, and they published some of the guidelines for pre-analytical conditions that I think are very valuable. And also we are working as the Society of Liquid Biopsy with the American Molecular Pathology to trying to get some, I will not say consensus because we don't have still rules, but at least some picture what is happening for the minimal requirement for liquid biopsies labs to work, because obviously we have a hole in the, I would say not legislation, but rules implementations. So for this reason, we see several variabilities in different areas of the world. And actually some of the regions in the world, I saw that they are selling, for example, exosome results. So that is really completely not acceptable when we have a not validation process. So I think we need to do a lot to make rules and try to make an harmonization between the technologies that we have.
Agreed entirely on that one.
Dr. Rolfo, can I ask you this question here? So somebody said they've read that some tumours are better ctDNA shedders than others, and I think we were aware of that, and also aware of exercise and all other different things that can potentially affect levels of ctDNA. Does this affect the clinical utility?
Yeah, so that's a problem in some patients. We call the non-shedders, that are patients that are really not having any alteration. And even we saw that in some patients that have a really big tumoral burden. So it's not depending on tumoral burden in some patients. We don't know what is that. In the beginning, we was putting all the blame in the technologies. Obviously, we was using Sanger sequencing. Is completely different from the high throughput next generation sequencing. But there is still patients that are completely non-shedder, and we see these differences between tissue positive and liquid biopsy negative. And that's a problem for monitoring these patients. So that's something that we need to reflect.
We have one question about, sorry, Dr. Rolfo, go on.
No, I was mentioning another thing that is interesting, and we are trying to implement in collaboration with the technology that Memorial is doing in the impact for CSF results. And that is when we have, and that happen in some patients who have only brain metastasis as a progression site, and we don't see nothing in the liquid biopsy. So the use of CSF seems to be open a door for customized patients treatment, specifically also because CSF, and you can confirm that is much more pure than the blood to analyze these mutations. So that is a future that we will see. Obviously, it's an invasive situation because we need an LP for the patients to have the results, but it's giving interesting point of view for treat the patients.
Yeah, yeah. We have one question about pre-analytical QC. What would be recommendations from both EQA perspective and from liquid biopsy perspective? Because obviously for liquid biopsy, it's very, very important. Are there any recommendations or standard ways to do it before it will go into analytical phase?
I think, Alex, we can't make recommendations for particular assays or way of doing things. Again, it goes back to the in-house validation in the laboratory that you know that your extraction and what you have generated cfDNA wise is going to be fit for purpose to get your result from the method that you're actually using. We know that when samples are moved between labs, they might work brilliantly in one laboratory where it's been validated, but not necessarily in the other laboratory when things are slightly different. So I think, you know, and as Simon says, the over 60 different methodologies, and that's not even looking at the extraction techniques on top of that. Who knows how many variables are out there? So I would say really is the general message is to make your in-house end-to-end pipeline work.
Yeah, pretty much use whatever you validated.
Yeah, exactly. Yes.
Yeah, if we can wrap up with one take-home message, we will say, if you don't, or my recommendation as an oncologist, I will say, if you don't have in your institution all the implementation or all the capacity to do next generation sequencing or testing or whatever you are using in the technologies, don't experiment with that. Try to create instead some networking with big institutions that they have this and the expertise, because it's not the same to do one every 3 months that have 100 every month. So it's important that we have this connection between the big centers and work together, because we are treating patients here. So that's the most important thing that we need to remember.
And I think that's where we see the critical interpretations coming through potentially as well. You need to keep up to date with the most recent information that's available in the trials, and really that comes with doing it all the time and not dipping in and out. So I would say that, yeah, again, I would echo that.
And I would add just onto that is that the EQAs is also, that's one of the things we're looking at. You know, we're trying to set clinical scenarios that challenge the knowledge of the laboratories and the scientists doing the testing and the writing the reports. And you can see from this year that, you know, we've included the KRAS G12C for that, exactly that reason. We need to see laboratories starting to consider all the options, not just fixed on one particular type of variant or type of test.
Absolutely.
Yeah. And we have one very technical question, I guess, to Dr. Rolfo. If I centrifuge the whole blood sample collected on EDTA tube and store the plasma at -80 degrees, how long cfDNA can be stable and usable for analysis?
I think I'm not the right person to answer that, but we are using, we are trying to use extract tubes because they are much more easy to handle. So there are several things, because sometimes as an oncologist, I say to my nurse, take the blood, and I know that if the blood is taken and I have the trust that the blood is going to the lab immediately, but we don't know what is happening between the blood is taken and the blood is arriving. So I don't know the temperature condition. I don't know how delay is arriving. So the best is try to use tubes that are like extract tubes that you can have even for 96 hours or 72, 96 hours. With the ADT tubes, we prefer to have an immediately process, at least a couple of hours maximum, but that is in our institution. So we are doing that. But I don't know if Sandi or Simon, if you have more experience in the part of the techniques, what is your experience on the EDT tubes?
You're absolutely spot on. That's generally what's happening here. I think what comes though with using Streck tubes in particular is the education that you don't put them in the fridge. You don't do what you normally do with a blood tube. So that adds another level of difficulty with implementation, but you know, that's not a blocker, and it does make a huge impact on the ctDNA that's extracted at the other end. So it's worth persevering, I would say. That's our experience in the UK anyway.
Great. Thank you very much for your participation, for your time, for great presentations, and thanks a lot for our participants for great questions and for the very, very useful discussion. And I guess with this, we will wrap up. Thank you very much, and looking forward for future events.
Thank you.
Thank you.
Thank you.
Thank you.